Hi, first of all, I'd like to thank the SBPZ Early Career Researchers Committee that is composed by Carolina Catapreta, Maria Fernanda Laranjeira Silva, and Sandra Maruyama, who invited me to speak here at the SBPZ Talks. Thank you very much. My name is Rubens. I'm a research scientist at Instituto René Rachou, which is part of Fundação Oswaldo Cruz in Brazil. And today I'm going to talk about the development of molecular diagnostics tools based on LAMP and nanotechnology to detect the DNA from Leishmania parasites. When we're talking about parasites, the microscopy is the technique used to see them. As we can see here, we're sure that these are Leishmania parasites within the macrophage and here in the bone marrow preparation. And this technique is very specific because we're seeing the parasites. However, this could be not sensitive enough to be considered alone for the diagnosis of leishmaniasis. We also have the LFD in that case, which is a antibody-based test, and it's very common during pandemics for the rapid test to detect the SARS-CoV-2 antigens for the COVID-19 pandemics. And the antibody test can also detect antibodies that are generated after an infection, for specifically talking about infectious disease. And we also have the molecular tests. Here I'm calling molecular the tests that are detecting a given RNA or a DNA. Normally they are more sensitive than the others, and they're considered the gold standard for a plethora of tests to detect pathogens, including infectious disease. And the ideal condition should be microscopy, antibody-based, and molecular together. And in this talk, I will focus only on molecular tests. Talking about pandemics, the PCR, or the polymerase chain reaction, as the gold standard for the detection of the RNA from SARS-CoV-2, this is now very well known. And normally the technique is associated with a centralization, with big chains of laboratories, and they need specifically equipment that normally are expensive, and they need also a very well trained personnel. However, we're now facing a huge technological advances. For example, we're sending people to space. So if you have money, we can go using a spaceship like this and spend some days taking picture of the Earth. And we are currently also very well connected. If we're talking about the satellite internet based, we can be connected in every spot on the globe. And what these advances has in common, they're allowing us to access technological solutions. For example, here we have the NASA astronaut Peggy Whitson, and she's running a PCR with this small thermocycler, or a PCR machine, call it mini PCR, in the International Space Station. So this is the decentralization of a molecular test. And normally this is high quality. So even comparable with these huge equipment that are used for high quality PCR diagnostics. And not only PCR, here also in this International Space Station, we can sequence genomes with this small machine here. And this is the real life, and this is reality, and is currently not in development, but people are using this in the International Space Station. This is our current time. So if you advance in such technologies, so why not to use this point of care approach during a medical appointment? For example, here we have a dog. We're talking about leishmaniasis, but this could be also applied for human disease. And thinking on the point of care diagnostics test, the World Health Organization created this acronym, the ASSURED, in which each letter corresponds to a characteristic, to a specific feature for the ideal point of care approach. So in that case, affordable by those at risk of infection, sensitive with very few false negatives, specific with very few false positives, user-friendly tests that are simple to perform and require minimal training, rapid to enable treatment at first visit, robust, for example, not requiring refrigerated storage, that would be the ideal condition, equipment free, and environmentally friendly. Normally, it's hard to have equipment free with high sensitivity, but the ideal condition should be at least one equipment of very small pieces and portable if possible, and delivered to those who need it most, right? And recently, a group of researchers added the RNE, and the acronym now is reassured, in which the R stands for real-time connectivity. This is because of the IoT, or the Internet of Things, that are connecting the device and could be also applied to this kind of tests, and also could help with automation. And the ease of specimen collection, this could be one of the main bottlenecks for the point of care diagnosis. This is called the pre-analytical phase, in which we have to access the target. And sometimes we need more technology to be easy to access these targets. And unlike the PCR method, which enables us to amplify the DNA to be detected, here I'm talking about the isothermal amplification methods that, unlike the PCR, we're amplifying the DNA in a specific range of temperature and not cycling like during the PCR. And here I'm going to focus on the LAMP, that stands for loop-mediated isothermal amplification. Here, it uses a DNA polymerase, and normally is the BST. This is because it's isolated from Bacillus staurotermophilus. It needs 4 primers that recognize 6 specific regions on the DNA. And normally we add two additional primers, the loop one, we'll see in a few minutes. And the temperature range is normally 6 to 65 degrees. And the reaction time could be in less than 1 hour. The target is normally DNA, but we can use as input the RNA that can suffer an RT, and after the reverse transcription, the DNA will be amplified. That's the technique that's being also used for COVID-19 diagnostics for the detection of RNA. There is a plenty of different techniques using different enzymes that works also in a specific range of temperature, but today we're focused on LAMP. That is one of the most known techniques. And currently, a startup from Germany checked the LAMP in peer-reviewed articles, and we're seeing here that the LAMP and the RPA technique, they contribute to 65% of the new knowledge in the papers, in scientific papers, are related to these 2 isothermal techniques, and the PCR is going down. And I'm gonna also to highlight here the CRISPR-Cas based diagnosis is also increasing. So sometimes to do the CRISPR-Cas, we need a pre-amplification step, and normally this is done using LAMP or RPA. So the LAMP is increasing in terms of numbers, and we are translating this knowledge to the society. So here are the molecular basis of the LAMP amplification. As you can see here, this F3 is the outer primer, the outer forward primer. The B3 is the outer backward primer. We have the internal primers here, the FIP and the BIP. And the characteristic that naming the technique, the loop, is these structures here. So after this dumbbell-like structure is formed, we have a cyclic amplification step. And after these, we have different size of product of amplification. So here are the 2 additional primers, the loop backward and the loop forward, that target the loop region. And this can increase the yield of the amplification products or the amplicons. So here we have the non-cyclic step, the cyclic phase, and the elongation. So depending on the size that is elongated, we have different size in the gel. So this is an example of, this is the marker, the negative control, and here is a positive for lamp reaction, in which band represent one amplicon in different size because of the technique, right? In talking about the primer design, today we have free available tools. Here's the lamp nap that is supported by New England Biolabs, and we have also the Primal Explorer, which is from the Icon Chemical Corporation. One of the inventors of the lamp technique is this company, and this is quite easy to design these primer sets, right? So here we can have the 4 conventional primers and the additional loop primers. In terms of lamp outputs, here, as you just saw, this is the smear that corresponds to the positive sample. This is the molecular size marker. We can also have a colorimetric output. So in these first 3 pictures here, we're seeing a colorimetric that is an output which is sensitive to the concentration of the magnesium in the medium. During the amplification, we have the precipitation of the magnesium pyrophosphate, and based on that, different sensors can be used, and what we have is this kind of pattern. As you can see here, this is negative and positive using the HNB or hydroxynaphthol blue. Here are malachite green, a mixture of other components. Those are positive and negatives. We have also the pH as a colorimetric sensing as well. The turbidimetry, fluorophore, as when we use the DNA intercalators, even by naked eye, we can see positive, which is different from the negative, and under UV light is easier to see as well. We can also have the output in real-time manner, right? So using specific fluorophores, we can see the amplification in real time, so in function of time. And we can also add these products of amplification in the presence of a LFD stick. And depending on the treatment, we can see the test band and the control band. For example, here we have our amplicon that is tagged with biotin and also with FITs. So after passing the conjugating pad with gold nanoparticles, the amplicon will be specifically identified in the test line, and the control line is here to show that the test is going well. So this is a second step of diagnostics. So after amplification, we can run and see the output by LFD or lateral flow device. We have also a test for lamp outputs that is considering also gold nanoparticles, but in that case, the gold nanoparticle is functionalized with a DNA probe that can be complementary to the target. So in that case, what we have after salt addition is a change in color. So here what we have is a stabilization of these gold nanoparticles, and we're still with these red or pink color. However, when we don't have the complementation, which means the absence of the target, what we have as a result is a precipitation of these gold nanoparticles, so they aggregates, and there is a change in color. And as you can see here, we can simple by absorbance see the differences in the positive and the negative samples. During the DNA amplification, we have also a release in protons. So, which means that when you have DNA amplification, will decrease the pH, and then we can have a pH sensor within the reaction, and the yellow ones will be the positive and the pink will be the negative samples. And currently, we'll also have the CRISPR-based detection. In that case, we'll have a Cas, or a CRISPR-associated nuclease, that after recognition of a specific target, they can cleave a reporter. Here is like the TACMan, where we have a quencher and a fluorophore. After be cleaved, they will shine the fluorescence of this specific fluorophore, and then we have a fluorescent output. So in this example, we have here the Cas13 that is targeting RNA, and this is very important for viruses, as for example, there are some tests being used for SARS-CoV-2 detection, and the Cas12 that is targeting the DNA specifically. So we can have the fluorescent output, and we can also combine with LFD using also CRISPR-Cas based technique. And then can be, as I showed, that can be combined. So the first step is the amplification, and this is the detection step. And it can be at the same time, depending on the Cas you're using. So the technique using the pH as an output, it's being used during the pandemics, and is very useful, sensitive and fast way. In agriculture, for detection of Wolbachia infected mosquito to inhibit the viral transmission, and has also been sent to space, in the International Space Station, as a proof of concept. So this is a very useful technique. There is another kind of very nice approach, also using fluorescence, that is the displacing probes. In that case, we put in the reaction for example, here's the loop backward that is with a new sequence here, and coupled with a fluorophore. So in the reaction, we put also the quencher with a complementary structure here that will keep without fluorescence. So since the primer recognizes the target in a reaction, the BST polymerase will start the DNA polymerization, and also they have a domain that will, a helicase domain that will take out this quencher here, and then we'll have a specific fluorescence. So when you have a colorimetric or a fluorimetric using DNA intercalation output, this is in specific. So every DNA that is being amplified will lead to a positive output. However, in that case, what we have is a fluorescence that is present only in the presence of the target. And here, as you can see, is an example from a paper from 2017 showing the detection of Zika, chikungunya, and dengue. Here is in the singleplex manner and in a multiplex. So together with the other primers that identify different viruses. Yes, there is a delay here in the time of detection. However, these can be easily used for multiplex purpose. And the technique was also useful for detection of the Zika virus in mosquitoes. So here the preparation was in a paper, and the paper was used for the lab reaction. And as you can see here, this is a positive output after using this technique. So the main advantage here is to multiplex different targets in the same tube. And after, based on the specific fluorescences, we can see different targets in the same reaction. And why this is important for Leishmania diagnosis, for Leishmaniasis diagnosis. So here we have a feline Leishmaniasis, and here in these pictures, we have the sporotrichosis, which is quite similar in terms of clinics. So using a molecular technique that can easily be accessed by a biopsies or a swab, and can be compatible with point of care characteristics, is very useful, for example, to differentiate and to avoid misdiagnosis of Leishmaniasis. This is an example of feline leishmaniasis. However, we have in Brazil and several parts of the world a visceral leishmaniasis, however, that they're being caused by dermatrophic species and vice versa. And that was based on that that we started a project. This was the project of the MSc student Ellen Gonçalves at FIOCRUZ, with the main goal of developing a LAMP assay to detect species-specific Leishmania. So what we did firstly was to collaborate with the bioinformatics people in Santiago in Chile. This was a work that was made by Dr. Vinicius and his PhD student Eduardo Martinez at the Universidad de Chile. And what they did was a pangenome. So they compared 26 different Leishmania genomes that was publicly available. And after that, they did a pangenome. So all the sequences that were shared were not interested on. We were interested on the accessory genome. And in this accessory genome, we picked up the specific sequence to design the primers for LAMP reaction. So what Ellen did was first to validate this set of primers. As we can see here, this is the primers that were designed for Leishmania amazonensis, amplifying only Leishmania amazonensis and not crossing with human DNA, or not crossing with Leishmania brasiliensis or Leishmania infantum DNA. And this is the negative control. And the same was seen for Leishmania brasiliensis and for Leishmania infantum. So after that, Ellen did the colorimetric output here with different concentrations of Leishmania infantum DNA, presenting here a limit of detection around 0.01 nanograms, which meets the clinical relevant concentrations of the DNA in blood or bone marrow samples, for example, to the diagnostics of visceral leishmaniasis. And she also mixed all the other primers in these tubes to see if the reaction will still positive. And as you can see here, they still positive even in the presence of the other primers, showing that the multiplexing or the other primers are not impairing the reaction, the specific reaction. In that case, Leishmania infantum, right? And after that, she did the displacing probe techniques. So in this case, each loop primer was tagged with a specific fluorophore. In that case, FAM from infantum, HEX for Leishmania amazonensis, and TAMR for Leishmania brasiliensis. And this is a single plex reaction showing that after 1 hour, she was able to detect the three species independently, however, in the same reaction at the same time, showing that LAMP multiplex using displacing probe techniques is feasible in this case. And she also did the multiplex, which means that she put all the probes and all the primers in the same tube. And as we can see here, there is a huge delay on the time. Here, 140 minutes, more than 2 hours to detect, the three species. So it's better to use the single plex reaction. However, we're here using different fluorophores, and we have to test if these gain are specific or not, depending on the target, or specific for the fluorophores, right? So after proving that we can use the displacing probes as a proof of concept for LAMP to the diagnostics of leishmaniasis, she tested these, or she validated using clinical samples. And as you can see here, she got a incredible 80, almost 90% sensitivity when using clinical samples from dogs, and this is blood and bone marrow, and I think it's lymph node also, that were used here for the DNA detection. And we have to keep in mind that these samples were they have the DNA extracted, So which means that the concentration of the DNA within the samples, the specific Leishmania DNA is very low. She did the same thing for Leishmania brasiliensis. This is using biopsies, and in that case, not from dogs, this is from humans. And she got a 60% sensitivity. Again, here you have to keep in mind the fluorescence gain. Here you have the background for the primer that is specific for the fluorophore that is specific for Leishmania infantum. And also in cutaneous leishmaniasis biopsies, we have a low concentration in Leishmania DNA if we compare to Leishmania infantum. So we have to optimize here the input to increase sensitivity. And we are now working with Alessandra, a PhD student who is continuing the Ellen's work. And she project is also the Leish ID. And what she did was to replace the hex and tamara fluorophores to detect Leishmania brasiliensis and the amazonensis here for only to use the FAM. And as we can see here, now we can correct and even have a very rapid test in only 20 minutes, being able to detect the 3 different species. So this is a single plex. However, again, this isn't the same reaction. So the 20 minutes is to detect specifically or species-specifically 3 different species of Leishmania. So this is very important to epidemiological surveys in regions in which one species is causing atypical leishmaniasis. And what Alessandra also did was the cross-reactivity test. So here we can see the primers designed for Leishmania amazonensis in the presence of the Leishmania amazonensis DNA. And we have a small cross reaction here with Leishmania mexicana, but this is normal because they're very similar and they can be part of the same complex. However, there is no cross reactivity with among the other Leishmania species. And she did the same with the primers for Leishmania brasiliensis, and not surprisingly, we're amplifying here Leishmania guianensis. And we think this is specific because also they're very similar. However, there is no cross-reactivity with the other species. And for Leishmania infantum, there is no cross-reaction with the other, all the other Leishmania test species. This was made in collaboration with Rodrigo Soares, one of our partners, Jerônimo Rugani and Felipe, who gave us the samples, the Leishmania samples. So thank you to them. And how can we translate what I'm talking about, the scientific basis to the communities, to the society? So thinking about that, our group in collaboration with a company here in Belo Horizonte, we developed the OmniLamp. The OmniLamp is a portable machine that is able to detect a given RNA or DNA based on LAMP and for the detection of given pathogens. So the machine is controlled by an app, and this app is also responsible for the input of samples that can use QR codes or barcodes, and the app will control all the temperature, all the reaction, and at the end of the reaction, the app can also interpret the results in an automatic manner. So we can have it very fastly in our hands after the time of the reaction. So for example, to detect Leishmania, as we just saw, in 20 minutes, we got the outputs already. So it can be also adapt to quantitate, but right now we're just using a qualitative manner. And how it works, the equipment is able to detect different colors. So in that case, the lamp reaction, in which using the pH sensors, in which the yellow color corresponds to the positive. And if we compare to other machines that are also using lamp-based techniques for the detection of pathogens or a given targets, the OmniLamp is the only with 12 slots. So here we have two space for the controls, the negative and the positive controls. And these couple samples are the test and the control. So we can have 5 coupled with the internal control, or we can have 10 samples without internal controls. And now we are currently working in another technique, which is coupling the LAMP technique together with nanotechnology. And this work is done by, it's been done by Dr. Ana Carolina Lage, Letícia Trindade, and Amanda Bonot. So Ana Carolina is a researcher also at René Rachou Institute. Letícia and Amanda, they are postdocs. Letícia working on virus and Leishmania, and Amanda also. So we started together working on the diagnosis of COVID, and based on that experience, we're now passing through and applying this to the detection of Leishmania. So the idea is to have the DNA that was amplified by LAMP, as I just showed. In the presence of this gold nanoparticle, what we have is a specific recognition of the probes that is in the biosensor. The biosensor here is the gold nanoparticle with the biosensor, and the positive reaction will stabilize this formulation. However, when you don't have the DNA or the specific target after a LAMP amplification, what we have is a precipitation. So these are the results of some preliminary results on the solution. As you can see here, is a Leishmania infantum specific probe, which is in the biosensor. And when we add, for example, 20 microliters of the amplicon that came from the LAMP reaction, we have a stabilization of the reaction here, which means this is precipitation under the DLS, or dynamic light scattering, and this is the stabilization. And we can see here easily the difference between the positive or in the presence of the Leishmania infantum amplified target, and also for Leishmania brasiliensis here and Leishmania amazonensis. So we're still developing these tests, but the idea is to have a tool that is quite easy to use on the field. For example, here we have, this is a real life experiment. So this is the positive, quite pink, and the negative results that could be colorimetric. So this is the LAMP reaction and the reaction after adding the biosensors. And this is very fast, like a few minutes, we can have these difference very easily. And the name of the test is YOUNG, and that stands for the isothermal amplification nanogold together. So we're still working on that to try to translate this knowledge to this society. So with this, I would like to thank the students that were involved in this project, the alumni, for example, Ellen, that worked on this project, my collaborators outside FIOCRUZ, and also my partners within FIOCRUZ, and of course the funding agencies. Without them, we're not able to advance science. So thank you very much. Bye-bye.